Good morning, student. In the last lecture, we were discussing about our first chapter in the paper number first, diversity of cryptogam. Okay, the paper it mainly deals with the plants which are not producing the seeds. Okay, the paper it is having the three unit. The first unit is dedicated to the microorganisms. The second unit it includes the study of fungi, and the third unit we will be discussing regarding the fungi. Okay, the first chapter, viruses. Okay, in the last lecture we were discussing the general characters of the viruses. Okay, these were the learning objectives which we have discussed in the last lecture. Then also virus it is derived from the venom, which means the poisonous. Okay, then also we have discussed the history of the study of the viruses. In the history of study of viruses we have discussed that V. Byjernik in the 1898 for the first time coined the term virus. He defined the virus as the infectious living fluid and demonstrated that they differ from the other cellular organism. We have discussed in detail regarding the history of the history of the study of the viruses, and also the general characteristics of the viruses. Viruses they cannot be defined in one line, but the viruses these can be described depending upon their characteristic features. Okay, depending upon their characteristic features we can say that the viruses these are obligatory parasites. Obligatory parasites, these are completely inert. These are not harmful when these are present outside the host cell, but once they invade, once they penetrate into the host cell, they will start to multiply. That's why we are saying that the viruses are obligatory parasites. Then self-replicating, the viruses can multiply themselves. That's why these are self-replicating, and these are non-cellular. Non-cellular we are saying that because the viruses these are not having the prokaryotic or the eukaryotic structure. The viruses simply these are made up of only two components, the centrally present genetic material. Genetic material, either DNA or RNA is present. In that one, the viruses these are completely different from the other cellular organisms. Okay, in the other cellular organisms what happens, the both DNA and RNA are present as a genetic material, but in viruses if you see, the viruses these are having only one type of the nucleic acid. Either these are having the DNA or the RNA. Okay, and these are non-cellular. The genetic material it is then well protected by the protein coat. Okay, thus the viruses these are non-cellular. That means these are not having the eukaryotic and prokaryotic structure. These are only made up of two components, the centrally present nucleic acid, and this nucleic acid genetic material it is protected by the protein coat. Okay, then we have also discussed the viruses sizes and the shapes. Then viral structure in the overview we have discussed. In the center you can see here there is a presence of the genetic material, either DNA or RNA, then which is well protected by the protein coat. That protein coat is called as the capsid. The capsid it is composed of many subunits, and each of the subunit is called as the capsomere. Then in some of the viruses, the protein coat it is also then covered by the additional layer. That additional layer is called as the envelope, and on the envelope there is a presence of the small projections, and these projections are called as the spikes. Okay, it is not necessary that all the viruses the protein coat it is again covered by the envelope. Okay, that means the viruses these are having only the nucleic acid and the protein coat. Envelope is entirely absent, and such a type of the viruses are referred as the non-enveloped or the naked viruses. Okay, then also we have discussed regarding the nucleic acid. Okay, just I told you that the viruses these are having only the one type of the nucleic acid. Okay, they completely differ from the other cellular organisms. Other cellular organisms, both DNA and RNA are present. Okay, but in viruses only DNA or RNA is present. Okay, we have discussed in detail regarding the nucleic acid in the viruses. Okay, and we were about to discuss the protein code. Okay, the protein code, central genetic core, central genetic core, it is well protected by the protein code. Okay, central genetic core, it is well protected by the protein code, and that protein code in the viruses, it is referred as the capsid. Then the capsid consists of several identical protein subunits. Okay, large number of the protein subunits come together and then they will form the capsid. Okay, these are several identical protein subunits. These are as the capsomeres. Okay, understand that the capsid, it consists the protein code surrounding the genetic material known as the capsid, and the capsid consists of several identical protein subunits, and these are known as the capsomere. Okay, then again the capsomeres made up of a single or many type of the proteins. Okay, in some viruses the composing the capsomeres are of a single type, while in the several types of the proteins may be present in the capsomere. ठीक आहे. जे प्रोटीन कोट असतं व्हायरसच्या भोवती, जेनेटिक मटेरियलच्या भोवती, त्याला आपण कॅप्सिड म्हणतो, आणि हे कॅप्सिड खूप सारे प्रोटीन सब युनिटनी बनलेलं असतं. त्या प्रत्येक सब युनिटला आपण कॅप्सोमियर्स असं म्हणत असतो. ओके, आणि हे कॅप्सोमियर काय एकाच प्रकारच्या प्रोटीनचं बनलेलं असेल किंवा खूप सारे प्रोटीन्स एकत्र येऊन ते कॅप्सोमियर बनलेलं असू शकतं. Okay, then usually these subunits are arranged, usually these protein subunit, or we can say usually these capsomeres, these are arranged in helical or polyhedral geometric form. Okay, these are usually arranged in helical or the polyhedral geometric form. We will discuss what is the helical and the polyhedral geometric form in the next slide. Okay, these capsomeres, just remember here, these capsomeres, these are usually arranged in helical or polyhedral geometric form. Okay, then again these capsomeres are of two types. Capsomeres, these are of two types. The first one is the pentamere and another one is the hexamere. Okay, capsomeres, these are made up of two types. The capsomere, these are of made up of two types, pentamer and the hexamer. What is mean by the pentamer and what is mean by the hexamer? The pentamer, it is made up of, penta means five, it is made up of five monomers, and the hexa means six, and the hexamer, it is made up of six monomers. Okay, the capsomeres are of two type. The capsomere which is made up of five identical monomers, then it is called as the pentamer, and when the capsomere, it is made up of six monomers, then it is said to be the hexamer. Okay, the each monomer, it is connected with the neighboring monomers on the either side with the help of the bonds. Okay, whatever may be the capsomere, it may be the pentamer or the hexamer, the monomers, these are connected with the neighboring monomer with the help of the bonds. Okay, and likewise the capsomeres, these are also connected with each other. Okay, the capsomeres, these are also connected with the each other, but the bonds between the capsomeres are weak. Okay, the monomers are also connected with each other with the help of the bonds, and again the capsomeres are also connected with each other with the bonds, but the bonds which are connecting the capsomeres are weak. Okay, the bonds which are connecting the capsomeres are weak. Okay, then again there are some complex forms of the viruses. Why these are the called as the complex form? Because the capsid protein coat, it is covered by the additional layer, and that additional layer is called as the envelope. Okay, some of the viruses, these are the complex forms. These are said to be the complex form because the capsid or the protein coat, it is covered by the envelope. Okay, that is why we are saying that these are the complex forms. Okay, in some viruses the envelope may have the projection called as the spikes. Okay, in the photograph, in the last lecture we have discussed, in the last slide we have discussed regarding the envelope and the spike. Okay, that envelope, the envelope which is surrounding the capsid, it is usually made up of some combinations of lipids, proteins, and carbohydrate. Okay, the envelope which is covering the protein coat, it is made up of combination of lipids, proteins, and carbohydrates. Okay, then the another one, the envelope of many viruses have the projections called as the spikes. Okay, again these spikes, these are made up of carbohydrate protein complex. ओके, लक्षात आलं. जे एन्व्हलप आहे, जे कॅप्सिडला ऍडिशनल लेअर म्हणून वर्क करतं, ते एन्व्हलप कशापासून बनलेलं असतं? तर तीन त्याच्यामध्ये कन्स्टिट्युएंट्स असतात. कार्बोहायड्रेट असतात, प्रोटीन असतात, आणि लिपिड असतात. ओके, आणि त्या एन्व्हलप वरती जे छोटे छोटे प्रोजेक्शन्स प्रोसेस्ड असतात, त्याला आपण स्पाइक्स म्हणत असतो. ते स्पाइक्स कशापासून बनलेले असतात? तर कार्बोहायड्रेट आणि प्रोटीन पासून बनलेले असतात. Okay, then what is the function of this envelope and the spikes? Okay, the viruses attach themselves to the host cell by means of spikes. Okay, that is the function of the spikes. With the help of the spikes, these viruses attach themselves to the host cell. Okay, that is the function of the spike. Okay, then beside the attachment, the spikes, these are the characteristic as an important tool for the identification of the viruses. Okay, the spikes, the main function of the spikes, the attachment of the viruses to the host cell, it is facilitated by the spikes, and spikes, they serve as an important tool for identification of the viruses. Okay, then again the viruses in which the capsid, it is not covered by the envelope, these are known to be the naked or non-enveloped viruses. ओके, ज्याच्यामध्ये हे एन्व्हलप अब्सेंट असतं, एन्व्हलप प्रेझेंट नसतं, त्याला आपण नेकेड किंवा नॉन एन्व्हलप्ड व्हायरसेस असं म्हणत असतो. ओके, लक्षात घ्या की एन्व्हलप हे सगळ्या व्हायरसेसमध्ये प्रेझेंट नसतं. काही काही व्हायरसेसमध्येच हे फक्त ऍडिशनल लेअर प्रेझेंट असतं. ज्यामध्ये ते प्रेझेंट नसतं, अशा प्रकारच्या व्हायरसेसला आपण नेकेड किंवा नॉन एन्व्हलप्ड व्हायरस असं म्हणत असतो. ओके, जसं एक्झाम्पल सांगायचं झालं तर टोबॅको मोझाईक व्हायरस. इन द टोबॅको मोझाईक व्हायरस, the capsid, it is not covered by the envelope. That's why the tobacco mosaic virus is naked or non-enveloped virus. Okay, then we have discussed the function of envelope and spikes, that is it facilitates the attachment to the host cell. But how the viruses which are not having the envelope and spikes, they get attached to the host cell? That is, in such type of the viruses, the capsid itself, the protein coat itself, facilitates the attachment of the virus to the host surface. Okay, when the envelope and spike is absent, in that case the protein coat or the capsid, it facilitates the attachment of the virus to the host surface, and it also protect the virus nucleic acid, virus genetic material from the nuclease enzyme present in the biological fluids. Okay, that protein coat, it facilitates itself for the attachment to the host cell, and it also protecting the genetic material of the virus from the nuclease enzyme which is present in the biological fluids. Okay, that is about the structure of the viruses. Okay, in the examination, if the short note is asked on the ultra structure of the virus, then you have to mention all these points and you have to draw the diagram of the virus. Okay, the diagrams are very essential while giving the answer in the examination. Okay, if you does not draw the diagram of that particular question, then your mark will be get reduced. Okay, remember that our paper botany, it is completely different from the other paper. The diagrams are very essential. The marks are allotted to the diagrams. Okay, that's why in each and every question, if the diagrams are necessary, you should draw the diagrams. Okay, separate marks are allotted to the well labeled diagram. Okay, that's why do not neglect the importance of the diagrams in our subject. Okay, then it is all about the structure of the viruses. Okay, then the general morphology, just you can see here the TMV. It is the tobacco mosaic virus. Here you can see the genetic material is present. That genetic material, it is surrounded by the protein coat. Okay, it is non-enveloped. Okay, additional envelope is not present in this particular type of virus. Okay, that's why it is called as the non-enveloped or the naked virus. Okay, then regarding the general morphology of the virus is that viruses, they may be classified into the various morphological types on the basis of their capsid architecture. ओके, म्हणजे त्याची कॅप्सिडची अरेंजमेंट कशी झालेली आहे त्याच्यावरून त्याच्या जनरल मॉर्फॉलॉजीवरती त्याचे वेगवेगळे क्लासेस केलेले आहेत व्हायरसचे. ओके, viruses, they may be classified into the various morphological types depending upon the capsid architecture. Okay, the structure of the capsid and individual capsomeres can be studied under electron microscope and X-ray crystallography. Okay, the viruses are very small. That's why if we have to study the structure of the capsid and the capsomeres, we have to observe the virus under the electron microscope and by using the technique X-ray crystallography. Okay, there are certain common morphological forms. Here we are discussing some of the common morphological forms of the viruses we are discussing here. The first one is the helical virus. Okay, here you can see the first one is the helical virus. Some of the common morphological classes we are discussing here. Some first one is the helical virus. Helical virus, here you can see this is the example of the helical virus. Helical viruses means what? These viruses are cylindrical or the simply rod like. Okay, the viruses which appears cylindrical or rod like, these are said to be the helical viruses. Okay, in this one you can see the central nucleic acid strand, it is coiled like a helical spring. Here in this photograph you can see here the RNA, it is the genetic material and which is coiled like a helical spring. ओके, एका स्प्रिंग सारखं हे कॉइलिंग झालेलं असतं जेनेटिक मटेरियलचं. Okay, and the capsomeres, these are also helically arranged around the helical springs. Okay, the capsomeres, these are also present helically around the helically arranged or helically coiled genetic material. Okay, for example, you can see the mumps virus, rabies virus and tobacco mosaic virus. These are the helical viruses. Example of the helical viruses: mumps virus, rabies virus, and tobacco mosaic virus. Okay, then the second morphological form, it is the polyhedral viruses. Okay, just first one we have discussed the helical viruses, in which the nucleic acid, it is present in the form, it is coiled and present in the helical form, and also the capsomeres and the capsid, which are present surrounding the genetic material, it is also present helically. Okay, the examples of the helical viruses are the mumps virus, rabies virus, and the tobacco mosaic virus. Then the second morphological form, here you can see the polyhedral viruses. Okay, here in this one, the polyhedral viruses, in the photograph you can see the nucleic acid, it is packed. Okay, here in this one you can see the nucleic acid, it is packed in unknown manner with a hollow polyhedral head. Okay, nucleic acid, it is packed. Here you can see this is the nucleic acid. Okay, this nucleic acid, it is packed in unknown manner within a hollow polyhedral head. Okay, then the polyhedral viruses, they may be tetrahedral, octohedral, and icosahedral. Okay, the polyhedral viruses, they may be tetrahedral, octahedral, and icosahedral, depending upon the number of phases. The polyhedral viruses, they may be tetrahedral, octahedral, and icosahedral, depending upon the number of phases. Okay, for example, in the icosahedral form, that is, it is having about 20 triangular faces. Okay, म्हणजे त्याला किती फेसेस आहेत त्याच्यावर डिपेंडिंग ते कोणत्या प्रकारचे व्हायरसेस आहेत ते ठरतं. दॅट इज दे मे बी टेट्राहेड्रल, octahedral, and icosahedral. Okay, for example, icosahedral in the icosahedral, it is having the 20 triangular faces. The examples of the polyhedral viruses are the adenoviruses and the polioviruses. Okay, example of the virus is the adenovirus and polio. Okay, then the next morphological form is the enveloped viruses. Just we have discussed the enveloped viruses. Okay, the capsid, it is covered by the specialized envelope. That envelope, it is made up of lipid, protein, and the carbohydrate. Okay, then these enveloped viruses, they may be spherical, helical, or polyhedral in shape. Example, HIV. Okay, human immunodeficiency virus. Okay, here the photograph of the enveloped virus. Here you can see the genetic material is present. This genetic material, it is well protected by the capsid. Okay, these are the small units of the capsids, which are referred as the capsomere, and here the envelope is present. Okay, the protein coat, it is covered by the envelope. Okay, these are the enveloped viruses. Okay, example of the enveloped viruses is HIV. Okay, then the complex type of viruses are present. Complex type of viruses, the example of the complex type of the viruses is the bacteriophages. Okay, phage means what? It is a Latin term. Phage means to eat. Okay, the viruses which are eating the bacteria. Okay, literally we can say the viruses which are eating the bacteria. Okay, but we can say the bacteriophages means what? The viruses which are infecting the bacterial cell. Okay, here this is the structure of the bacteriophages. Okay, it is very complex. Okay, these are said to be the complex viruses because these are having such a polyhedral head. Okay, in the center of this head there is a presence of the genetic material, and this head, it is then well protected by the protein coat. Then here you can see the tail like structure, elongated structure is present, and that tail, it is attaches to the head region with the help of the neck like region. Okay, that means there is a presence of the head, neck, and the tail. Again, this tail region, it is well protected by the protein coat. Okay, that at the end of the tail sheath there is a presence of the tail plate. Okay, or it is in this photograph you can see it is mentioned as the end plate. Okay, this at the tail plate there is a presence of the small pin-like structures. Okay, small pin structures, and these are called as the tail fibers. Okay, the function of the tail plate or the end plate and the tail fiber is to attach the bacteria, to attach the virus, sorry, to attach that particular virus to the bacterial cell. Okay, with the help of the tail plate or the end plate and the tail fibers, this virus it get attached itself to the bacterial cell. ओके, लक्षात आलं कॉम्प्लेक्स व्हायरस कशामुळे म्हणतात? कारण याच्यामध्ये वेगवेगळे रिजन्स प्रेझेंट असतात. म्हणजे काय, इथं हेड रिजन आहे, त्या हेड रिजन मध्ये सेंटर प्लेसला जेनेटिक मटेरियल आहे, या जेनेटिक मटेरियल भोवती प्रोटीन कोट प्रेझेंट आहे, नंतर एलॉंगेटेड पोर्शन प्रेझेंट आहे, त्याला आपण टेल रिजन म्हणतो. हे टेल रिजन आणि हेड रिजन एकमेकाला कशानं अटॅच झालेलं आहेत? तर नेकनं. ठीक आहे, आणि ह्या टेल रिजनच्या एका एंडला एक प्लेट लाईक स्ट्रक्चर प्रेझेंट असतं. त्या प्लेट लाईक स्ट्रक्चरला आपण टेल प्लेट किंवा एंड प्लेट असं म्हणत असतो. त्या एंड प्लेट पासून असे फायबर किंवा पिन स्ट्रक्चर्स प्रेझेंट असतात, त्याला आपण टेल फायबर्स किंवा टेल पिन्स असं म्हणत असतो. ओके, ह्या टेल प्लेटचं आणि ह्या टेल फायबर्सचं काय फंक्शन आहे? तर एखाद्या बॅक्टेरियल सेलला जेव्हा अटॅच होतात तेव्हा हे टेल प्लेट आणि टेल फायबर त्या अटॅचमेंटला त्या व्हायरससाठी त्यांना हेल्प करत असतात. ओके, दॅट इज these are the four most important morphological forms of the viruses. Okay, the first one is the helical virus, second one polyhedral virus, third one enveloped virus, and the fourth one is the complex virus. चार मॉर्फॉलॉजिकल फॉर्म्स आपण डिस्कस केलेले आहेत. ओके, चार वेगवेगळे मॉर्फॉलॉजिकल फॉर्म्स आपण व्हायरसचे डिस्कस केलेले आहेत. त्याच्यामध्ये पहिला जो होता तो होता हेलिकल फॉर्म, दुसरा पॉलिहेड्रल फॉर्म, तिसरा एन्व्हलप्ड फॉर्म आणि चौथा तो म्हणजे कॉम्प्लेक्स व्हायरसेस. ओके, that is all about the morphological forms of the viruses. Okay, then the next one, the classification based on the host. Okay, classification based on the host. In our syllabus, the classification based on the host, it is included. Okay, classification based on the host means what type of the cell it is infected by that particular virus. Depending upon that, the viruses are classified. Okay, that is, there are certain specific viruses which are infecting only the plant cell, or the specific or the particular type of the viruses which are infecting only the animal cell, and there are certain type of the viruses which are infecting only the bacterial cell. Depending upon the host, these are classified. Here you can see that depending upon their host range, which type of the cell it is infected by the virus, and clinical, epidemiological, and pathological symptoms. Earlier, the viruses were classified into the four groups. The first one was the plant virus. Plant virus means what? The viruses which are infecting only the plant cell. Okay, then second one, invertebrate viruses, the viruses which are infecting only the cells of the invertebrate animals. Okay, then vertebrate viruses, this group includes the viruses which are infecting the vertebrate animals or the cells of the vertebrate animals. And the last one here you can see the dual host viruses. Dual host viruses, that means the viruses which are infecting the both invertebrate animals and the vertebrate animals. These are included in the dual host viruses. Dual host viruses, this group includes those viruses which are infecting the two different hosts. Okay, two different hosts. That means these are infecting the invertebrate animals and the vertebrate animals. Okay, here in the plant viruses, you can see all the viruses which are infecting the algae, then fungi, and also the viruses which are infecting the bacteria. These are all included into the plant viruses. Okay, remember that the viruses which are infecting the bacteria, then algae, then fungi, then higher plants, all these viruses, these are included in the plant viruses earlier. Okay, but now the Holmes in the 1948, he classified the viruses into the three groups. Okay, according to Holmes in the 1948, the viruses, these are classified into the three groups. The first one is the phagini. Here the term phagini means what? Phage. Just we have discussed that phage means the to eat. Phage, phage. The meaning of the phage, it is a Latin word that means to eat. Okay, that's why the terminology it is phage here used. According to Holmes, he included all the viruses in a single order, virales. Virales. Okay, Holmes in the 1948 included all viruses in a single order, virales, and these this order virales, it is divided into the suborders. Okay, the order virales, it is divided into the three suborders. The first one is the phagini. This suborder includes viruses infecting the bacteria. Okay, phagini, it includes the suborder phagini, and this suborder includes the viruses which are infecting the bacteria. म्हणजे जे बॅक्टेरियोफेजेस आहेत किंवा जे व्हायरसेस फक्त बॅक्टेरियल सेलला इन्फेक्ट करतात ते सगळे फॅजिनी मध्ये इन्फेक्ट केलेले आहेत. Okay, then the second one, it is the phytophagini. Phyto means the plant, that means the viruses which are infecting the plants, these are included into the phytophagini. Okay, and the third suborder, it is the zoophagini. Zoo means animals. Okay, that means the viruses which are infecting the different animals, all these are included in the suborder zoophagini. Okay, thus according to Holmes, he included all the viruses into the single order, virales. The virales, it is then divided into the three suborders: phagini, the viruses which are infecting the bacterial cell; phytophagini, the viruses which are infecting the plants; and the zoophagini, the viruses which are infecting the animals. Okay, it's all about the classification of viruses based on the host. Okay, the question may be asked as a short note in the examination: classification of viruses based on the host. Okay, that is all about the classification based on the host. Okay, remember that in our syllabus only the classification of virus based on the host is included. Okay, then there are various means of classifications of viruses. Okay, but here in our syllabus only the classification which is based on the host is included. Okay, then the structure of the tobacco mosaic virus. Structure of the tobacco mosaic virus. Here earlier we have discussed that the there are various types of the viruses. These are discovered. These are discovered up till now. Okay, more than 100 types of the viruses. These are known to the human being, which causes the various diseases in the plants. Okay, we are discussing here only the viruses which are infecting the plants. Okay, up till now more than different types of the viruses, these are known, which causes the various diseases in the plants. Okay, of these the tobacco mosaic virus, the tobacco mosaic virus has been studied most extensively in the field as well as in the laboratory. Okay, more than 100 types of the plant viruses are discovered, but the tobacco mosaic virus, it is the virus which is extensively studied in the laboratory as well as in the field. Okay, this virus, it was firstly discovered, it was firstly reported by the D.J. Ivanovsky. Okay, D. Ivanovsky in the 1892. and in the crystallized form, that is an isolation and crystallization for the first time it was done by the W.N. Stanley in the 1935. Okay, by the W.M. Stanley in the 1935. Okay, tobacco mosaic virus, it was the first virus which was isolated and crystallized in the lab. ओके, सगळ्यात पहिल्यांदा रिपोर्ट झालेला व्हायरस कोणता आहे, तर तो आहे टोबॅको मोझाईक व्हायरस. आणि सगळ्यात पहिल्यांदा लॅबोरेटरीमध्ये त्याचं प्लांट पासून आयसोलेशन करून त्याचा प्युअर फॉर्म मध्ये त्याचं क्रिस्टलायझेशन करण्याचं क्रेडिट कोणाला जातं, तर W.M. स्टॅनले इन द 1935. And for his extraordinary work, he was awarded with the Nobel Prize. He was honored with the Nobel Prize in the 1946. ओके, सगळ्यात पहिला व्हायरस हा डिस्कवर झाला टोबॅको मोझाईक व्हायरस. याचा सगळ्यात पहिल्यांदा रिपोर्ट केला डी इव्हॅनोस्कीने 1892. ओके, आणि त्याचं प्युअर फॉर्म मध्ये आयसोलेशनला खूप सारं वर्ष जावं लागलं. 1935 मध्ये सगळ्यात पहिल्यांदा W.M. स्टॅनलीनं त्याला लॅबोरेटरीमध्ये प्युअर फॉर्म मध्ये आयसोलेट करून त्याचं क्रिस्टलायझेशन केलं. आणि त्याच्यामुळे W.N. स्टॅनलीला 1946 मध्ये नोबेल प्राइजनं त्याला सन्मानित करण्यात आलं. Okay, this virus here you can see it is a helical type of virus, just we have discussed. The virus, it appears as a rod or needle shaped. Okay, the virus, it appears as a bundle of rods or the needles. Okay, it appears here, you can see the bundle of small rods or the needles. Okay, it appears as a bundle of rods or the needles under the electron microscope. Then the each rod is approximately 3000 lambda in length. Each rod, it is approximately 3000 lambda in length and 170 lambda in the diameter. Okay, 3000 lambda in the length and 170 lambda in diameter. Then it has a molecular weight of approximately 4 lakh. Okay, molecular weight of the tobacco mosaic virus, it is about 4 lakhs. Okay, then the electron microscope and X-ray crystallographic investigations have revealed that these particles have the two constituents. Okay, when the tobacco mosaic virus, it was observed under the electron microscope with the help of the X-ray crystallography, then it was revealed that the virus, it is made up of only two components: centrally helically present nucleic acid, and this nucleic acid which is surrounded by the protein coat. Okay, it is a simple virus, it is not having the additional layer which is called as the envelope. Okay, the electron microscope and the X-ray crystallographic studies, it was revealed that the tobacco mosaic virus, it is made up of only two components, that is the protein coat and the nucleic acid. Okay, then here you can see the protein coat, this is the protein coat. Okay, this protein coat, it is made up of several subunits. Okay, these are the subunits of the capsid, and these subunits of the capsid are known as the capsomeres. Okay, the protein coat, it is made up of about 2130 identical protein subunits which are called as the कॅप्सोमियर्स. ओके, म्हणजे हे किती सब युनिट्स आहेत त्या प्रोटीन कोटमध्ये, त्या कॅप्सिडमध्ये, तर 2130 सब युनिट्स आहेत आयडेंटिकल प्रोटीन पासून बनलेले, आणि त्या प्रोटीन सब युनिटला आपण कॅप्सोमियर असं म्हणत असतो. ओके, देन each of the capsomere again, it is made up of 150 long chain of amino acids and its molecular weight is 18,000. ओके, एका कॅप्सूलमध्ये 158 अमिनो ऍसिड्स असतात. ओके, आणि त्याचं मॉलिक्युलर वेट किती असतं, तर 18,000 असतं. Each capsomere consists of 158 long chain of amino acids and its molecular weight is 18,000. Okay, then the capsomeres, these are arranged helically around a central single stranded RNA. Okay, here you can see in the tobacco mosaic viral, द RNA इट इज द जेनेटिक मटेरियल. ओके, ह्याच्यामध्ये DNA प्रेझेंट नसतो, ह्याच्यामध्ये काय तर RNA असतो. दॅट मीन्स द टोबॅको मोझाईक व्हायरस इट इज द रायबो व्हायरस. ओके, time is running out. I think this is enough for today, because you may have the next lecture. Okay, that's why I'm stopping the sharing. ऐकू आलं का सगळ्यांना?
हो सर.
आणि पॉवर पॉइंट दिसलं का व्यवस्थित?
हो सर.
पॉवर पॉइंट दिसलं का?
हो.
पॉवर